Hello, and welcome to the Lunula Scientific Seminar. My name is Ryan Maloney, and I'm the Chief Research Officer for Prim Cogent Solutions. And today, I will be discussing the clinical science that has validated the Lunula for the treatment of onychomycosis. Onychomycosis represents the fungal infection of the nail. It actually affects, specifically, the nail bed and nail plate, completely covering the surface of the toenail. The infectious agent permeates the keratin layers of the nail plate, resulting in discoloration, disfigurement, pressure, and ultimately pain. It is estimated today that onychomycosis affects nearly 10% of the world population. That is roughly 31 million individuals in America, 86 million individuals in Europe alone. Because of this tremendous prevalence, Medicare, or healthcare firms, are responsible for nearly $43 million annually to simply treat onychomycosis. In the United States alone, nearly $250 million was spent roughly on nail debridement. It is representing a tremendous financial and healthcare resource burden, and this is shared throughout most Western cultures. The common causes of onychomycosis is resulting from dermatophytes. Dermatophytes represent 90% of reported mycotic cases. Specifically, they result in distal lateral subungual onychomycosis. Dermatophyte species include Tryptophyton rubrum or Tryptophyton metacragophytes. These represent 90% of the reported infections, with other types of non-dermatophytic infectious agents resulting in less than 10%. Most individuals today view onychomycosis as simply a cosmetic disorder. However, due to the hyperkeratosis or thickening of the nail plate, this results in other comorbidities, including emotional stress, experience of pain due to the increased pressure, which ultimately results in limited mobility. Moreover, onychomycosis is the result, or can result in, nearly a threefold higher risk of amputation if the patient is diabetic. Therefore, this is a medical disorder and requires a serious and valid form of intervention. Mainstay therapies include several types of anti-fungal applications, topical or oral. A majority of cases are treated with oral anti-fungal medications, and here we see a list of numerous or common therapies utilized today. Most oral antifungal medications are greeted with limited success, ranging from, as evident here, 6% to 60% success rate. Moreover, the treatment time required can range from simply 12 weeks to nearly 78 weeks of therapy. And with the best reported efficacy at 60%, that is not a viable solution for the treatment of onychomycosis. Oral antifungal medications have limited success because they're attempting to treat the most distal end of the body, systemically. Introducing a drug traveling through the blood system can fail to reach the fungal pathogen because the most distal extremity, the foot, may have limited blood flow. Additionally, oral antifungal medications are metabolized by the liver, and numerous studies have identified this metabolism can cause great strain on the liver, resulting in hepatotoxicity. There have been several cases of liver transplant when individuals were consuming oral antifungal medications. Accordingly, in the United States and throughout Europe, most patients must undergo liver function tests at baseline and weeks 4 and 6 to ensure that there are no alterations or negative impacts on the liver function. Additionally, individuals may not be able or qualify for oral antifungal medications simply because of the health risks that can transpire with consumption of this very volatile and harsh medication. Enter Lunula, a safe and effective technology that is a multifaceted approach for the treatment of onychomycosis. This unique technology applies two specific and clinically proven wavelengths, 635 nanometers and 405 nanometers. It delivers these two wavelengths utilizing a patented rotating line-generated beam, which ensures substantial surface area treatment as well as depth of penetration into the mycotic nail. The intensity ranges from 17.5 to 25 milliwatts, positioning this laser as what is known as a cold laser. The overall dosage applied is roughly 0.95 joules per centimeter squared. It is a portable, lightweight technology that can utilize numerous power sources, and its convenient use and its subtle therapeutic approach allows Lunula to be the only multifaceted approach on the market today. The Lunula treatment consists of just 2 treatments separated by 1 week, with each treatment roughly 12 minutes. When the patient enters the clinic, they are advised to take off shoes, socks, clean their foot if debris is present. Simply slip the foot into the Lunula technology. The technician assures that the foot is properly aligned, the device is activated, and the technician can now exit the room while the patient receives a comfortable treatment for just 12 minutes, treating all 5 toes along the extremity. The mechanism of Lunula is opposite or opposes a majority of lasers that have inundated or saturated the market. Most lasers on the market today utilize a photothermal mechanism. Simply put, it is the use of light energy to heat the tissue. This is for the purpose of protein denaturation, to elicit inflammatory response, to promote a wound healing response, or induce collagen remodeling. So the purpose of this type of laser is to introduce photonic or light energy to heat the tissue. Lunula is different. Lunula utilizes photochemistry as its core mechanism. Before we can discuss in great detail how Lunula operates and its mechanism of action, let's define very quickly some key terms of photochemistry. Photochemistry is the use of light energy to modulate or alter biochemical and chemical reactions. This is done by targeting a photoreceptor, a molecular entity capable of absorbing light at very specific and discrete wavelengths, and wavelength is simply the color of light. Photochemistry proceeds in 2 steps. The first is the primary response. This is the simple absorption of light via the photoreceptor. Now, it's the functional change and the overall behavioral change of the photoreceptor that engenders or triggers the secondary response within the cell. In this illustration, we can see or map out the mechanism utilized by photochemistry, and hence Lunula. We start by introducing a specific wavelength, and by introducing this wavelength, we are able to isolate or specifically treat a photoreceptor. By stimulating that photoreceptor, we change its overall function and behavior. That then sends a signal rippling or resonating throughout the entire cell, changing other enzymes and other proteins until an observable clinical effect is observed. The subtle approach of photochemistry is similar to that of an agonist effect of a drug. Now, we are all familiar with the agonist effect of a drug. When we introduce a drug, we are essentially introducing a molecular component into the body. That molecular component travels throughout the body seeking out a specific target, a receptor. Once it binds to that receptor, a secondary cascade or signal is triggered throughout the entire cell, affecting numerous proteins and enzymes, overall changing the very functionality and behavior of that impaired cell. We don't feel or know that the agonist effect of a drug is working. What we perceive is the overall benefit. The Lunula laser operates in a similar fashion. We don't feel or know the Lunula laser is successfully treating onychomycosis. What we perceive, however, in 3 to 6 months is a clearer, healthier nail. Lunula targets specific photoreceptors, just like the agonist effect of a drug would target different drugs or different receptors depending on the drug. There are 2 primary photoreceptors that are found in our human cells that Lunula successfully targets. The first is cytochrome C oxidase. This is an enzyme found in the respiratory chain of the mitochondria. Simply put, the power plant of the cell. The second is NADPH oxidase. This is an enzyme that's bound to the cell's outer membrane. Stimulation of these photoreceptors is done by introducing a specific or discrete wavelength. We are able to change or alter the very functionality of these receptors by introducing those wavelengths. And we are able to now affect the entire cell function simply by stimulating these receptors. Let's discuss in greater detail cytochrome C oxidase and its activation via the 635 nanometer wavelength. Cytochrome C oxidase is an important enzyme that facilitates the production of adenosine triphosphate, ATP, and reactive oxygen species, ROS. Both ATP and ROS catalyze specific cellular reactions within the cell that affect its overall behavior and function. Now, cytochrome C oxidase, or CCO, has a peak absorption at 635 nanometers. Therefore, by introducing 635, we are able to increase its overall production of ATP and ROS. So the activation of CCO proceeds by introducing 635 nanometers, giving Lunula the opportunity to upregulate ATP and ROS production. Now, when discussing NADPH oxidase, or NOx for short, we see that it is stimulated via the 405 nanometer wavelength. Now, quickly, NOx is an important membrane-bound enzyme that is responsible for synthesizing reactive oxygen species. Now, once ROS is produced, it is quickly converted into hydrogen peroxide, or H2O2. Hydrogen peroxide is very toxic for cells, including pathogens like bacteria or fungi. What proceeds is H2O2's breakdown of the protective cell wall and cell membrane of these pathogens. Therefore, by introducing 405 nanometers, the peak absorption of NOx, Lunula is able to increase the production of ROS, which results in the increased production of hydrogen peroxide, therefore increasing the breakdown of the pathogen cell wall and cell membrane. Overall, Lunula is able to alter the function of NADPH oxidase, or NOx, by introducing the 405 nanometer wavelength, increasing its production of ROS, which is converted into hydrogen peroxide, which begins to break down the fungal pathogen cell wall and membrane, weakening its defenses. Here we see a basic animation of Lunula's mechanism of action. We can see the dark coloration of the mycotic nail at the top, and when we begin to introduce the 405 and 635 nanometer wavelengths, the 635 penetrates down to the germinal matrix, treating down to the nail bed, while the 405 nanometer wavelength successfully treats along the nail plate and down to the nail bed. What results is the weakening of the mycotic agent's defenses by altering the production of hydrogen peroxide and breaking down the cell wall and cell membrane, while at the same time the 635 nanometer wavelength is able to upregulate ATP and ROS, increasing blood flow so that we can now have better infiltration of immune cells, nutrient and oxygen delivery. Overall, what this animation should represent is the multifaceted approach of Lunula, not just treating the fungal pathogen, but also using the body's natural defenses and resources to further attack and degrade the onychomycosis pathogen. The mechanism of action just described is well validated within the clinical literature. We see here a representation of the clinical support for the 405 and 635 nanometer wavelengths. 405 nanometers ability to successfully degrade the cell wall, cell membrane by upregulating the production of reactive oxygen species and hydrogen peroxide has been studied since 1939, and more recent studies in 2005 and 2012. The effect of 635 nanometers to increase blood flow, to help support our immune cells' functionality to destroy foreign invaders like the mycotic pathogen, and increase oxygen and nutrient delivery has been validated across numerous peer-reviewed publications in highly respected journals like Lasers in Surgery and Medicine. So overall, Lunula is backed by a solid and sturdy clinical foundation. Now let's walk slowly through the overall mechanism of action for Lunula. And in order to do this, we must break down each individual wavelength. The first wavelength we'll discuss is 405 nanometers. 405 nanometers is introduced and begins to stimulate NOx, or NADPH oxidase. Upon stimulation, it increases the production of reactive oxygen species, which is quickly and readily converted into hydrogen peroxide. By converting into hydrogen peroxide within the fungal pathogen, hydrogen peroxide becomes cytotoxic, starting to break down the fungal pathogen cell wall and membrane. This in turn weakens the pathogen's immune defenses and begins to kill or ultimately destroy the pathogen itself. Now, let's begin to analyze and look at the 635 nanometer wavelength. As 405 is weakening the immune defenses of the fungal pathogen, 635 at the same time is stimulating cytochrome C oxidase. By stimulating cytochrome C oxidase, we upregulate the production of ATP and ROS. Now let's look at individually both ATP and ROS, and let's start by looking at ATP. When ATP is increased, it activates the nitric oxide synthase pathway, both within immune cells, which is known as iNOS, and endothelial cells, which is known as eNOS. By activating the nitric oxide synthase pathway, we upregulate or increase the production of nitric oxide. Nitric oxide rapidly diffuses through the membrane or exits the cell and begins to relax nearby vasculature, resulting in an increased blood flow. This vasodilation is now going to deliver more oxygen, nutrients, and immune cells to the area. Now, by looking at ROS, we can see a completely different cascade emerge. ROS within immune cells is quickly converted into hydrogen peroxide, and that is important for immune cells as immune cells kill foreign invaders through the utilization of hydrogen peroxide. So as more immune cells are being delivered to the area, we're enhancing their ability to produce hydrogen peroxide and thus begin to break down and attack the fungal agent. Collectively, by increasing blood flow through vasodilation, increasing the delivery of immune cells, and by improving the overall function and behavior of the immune cells, we now have a natural attack to degrade and kill the fungal pathogen. This simultaneous approach, delivering 405 and 635 nanometers, allows Lunula to be the only multifaceted approach on the market today. The concurrent delivery of 405 and 635 allows Lunula to weaken the immune defenses of the fungal pathogen and at the same time increase blood flow, oxygen and nutrient delivery for faster nail growth, but also immune cells, and stimulating their function so that they can begin to break down the fungal pathogen, which was weakened by the 405 nanometer wavelength. Overall, this multifaceted approach allows Lunula to effectively kill the mycotic agent, increasing the blood flow and ultimately increasing the overall rate of growth of the nail. And this treatment is going to cover the entire nail plate, nail bed, and even deep as the germinal matrix layer of all toes. To understand the clinical utility of the Lunula laser, we must begin analyzing the in vitro studies that have been performed. Here we see a published study in 2011 that looked at the overall effect of 405 nanometers to upregulate reactive oxygen species. We see in the image on the left is a control sample. The control sample has been stained to look at the intensity or concentration of reactive oxygen species. When we compare that with a section or sample that was treated with blue light, we see a much more intense image. This is because the study confirmed that 405 nanometers upregulated the production of reactive oxygen species and therefore evident in the more intense staining in this particular slide. In another study, we see the clinical benefit of 405 nanometers to upregulate reactive oxygen species. This study, published in 2012, assessed numerous wavelengths. This particular graph compares red light versus blue light, or 405 nanometers. We see that red light produced a far smaller concentration of reactive oxygen species compared to 405 nanometers. Therefore, when attempting to eradicate the onychomycosis causing fungal pathogen with reactive oxygen species, the best wavelength to use is the one that's found in Lunula, 405 nanometers, which produces the greatest amount of reactive oxygen species compared with any other wavelength within the visible spectrum. We have seen that the 405 nanometer wavelength successfully upregulates the production of reactive oxygen species. In this particular slide, we identify the key benefits of 635 nanometers. We are looking at real-time Doppler ultrasound. In the upper image, we see a toe deprived of substantial blood flow. We see the white line indicating of the nail plate extending out to the hyponychium, towards the eponychium, and the germinal matrix layer. There is an insufficient amount of blood within this structure. Subsequent to Lunula, we see a tremendous increase or surge in the blood flow. This ultimately yields greater nutrient delivery for tissue regeneration, increased oxygen and immune cell transportation, and ultimately improvement in fungal cell death. Now, understanding the principles of 635 nanometers and 405, let's compare Lunula with other laser-based technologies that have entered the market. First, Lunula is the only technology that combines 635 nanometers and 405. Most other devices utilize a wavelength between 870 and 1320 nanometers. Lunula is also a non-thermal mechanism. Other devices, however, utilize a photothermal mechanism to yield their desired clinical response. This ultimately requires that they make physical contact with the toe, whereas Lunula does not require any physical contact made to the toe, nor does it require debridement of the nails. Most other technologies, because they are using heat, require debridement of the nail to get closer and closer and deeper down to where the colonization of the fungal pathogen resides. Now, because of this concentration of laser energy for the generation of heat, they can only deliver a small spot size. Compare the spot size here of 2 to 10 millimeters with the spot size or treatment size of Lunula, which is 28.62 centimeters squared. This ensures complete, effective treatment coverage of the nail plate, the nail bed, and surrounding tissue. The treatment time for Lunula is 12 minutes for all 5 toes on one extremity. Compare this with other technologies, requiring 4.5 minutes per toe. Accordingly, if more than 1, 2, or perhaps 3 toes are infected with mycosis, the treatment time can begin to extend well beyond 30 minutes. This does not include physical debridement of the nail, which can now increase the length of time to well above 60 minutes. Lunula has been tested with no nail debridement, keeping the treatment consistently at 12 minutes. The study performed with the Lunula Laser required just 2 treatments, separated by 1 week, with other devices on the market presently requiring at least 4 treatments. Furthermore, the clinical studies to evaluate Lunula did not include the application of a topical anti-fungal solution. Other technologies, however, required the use of a topical solution during the clinical trial. This raises the question, what component, either laser or topical solution, was responsible for the clinical change in toenail appearance? With no topical in our clinical trial, Lunula and Lunula alone was the causing factor for nail clarity. Looking at the studies, toes studied in our FDA clinical trial was 105, compared to 17 in most other trials, to some trials actually not even completed for other products. The mean millimeter change in new, healthy, clear nail for Lunula was 4.99 millimeters in 3 months. What this meant ultimately was that new nail grew out to a length of almost 5 millimeters in just 3 short months. The best clinical outcome reported for competitive technologies, 3.7 millimeters in 3 months. Now, what was the percentage of subjects actually yielding an improvement in nail clarity? By 3 months' time, 97% of subjects in the Lunula study saw some type of improvement in nail clarity, compared to just 71.4% for other devices. Let's take a quick look at really the discrepancy of surface area treatment. Let's look at a thermal-based laser, which represents a majority of other laser-based technologies on the market. Here we see small spot size by small spot size. Imagine moving this distance of 5 millimeters by 5 millimeters across the entire nail plate. Missing one small section could result in preservation of the fungal pathogen, which would allow for or permit recolonization of the nail plate. So, requirement regarding meticulous placement of each stimulation along the nail plate is required. When we compare this now with Lunula, it provides complete and total coverage of not only the nail plate, but the entire extremity or toe itself. This ensures that every aspect of the infected toe is receiving adequate stimulation, even deep down to the germinal matrix layer, eradicating the fungal pathogen and stimulating blood flow, oxygen flow, as well as increased infiltration of immune cells. Let's begin to understand the true clinical utility of Lunula by diving deep into the clinical trials that have been performed. The first clinical trial performed was an independent pilot investigation. It examined 168 toes across 3 independent clinical sites in the United States. A single 12-minute treatment was performed for all infected toes. No nail debridement was performed. In addition to no nail debridement, no anti-fungal topical was directly applied to the toes. Instead, it was applied to outside surfaces such as the shoes, socks, or other wet or moist surfaces such as a shower stall. This was simply to prevent or mitigate the risk of reinfection. Moreover, in this pilot investigation, no adjunctive modalities were used, just simply the application of Lunula. Patients were followed up between 9 to 72 weeks. This was a pilot investigation, therefore, the follow-up or protocol details were not rigorously defined. That was to occur later in the FDA study. This was to demonstrate at a very cursory level or superficial level the basic utility of Lunula. The outcomes observed in this clinical trial are evident in these before and after images. Here we see a toe with basic 30% infection along the nail plate. This is highlighted by the discoloration of the upper images. After the Lunula single treatment, 4 months later, the independent clinicians were able to document a 100% clear nail, indicating the clinical utility or effectiveness of the Lunula device. This pilot investigation provided substantial evidence that warranted the completion of a more rigorous and better defined FDA clinical study. The second image from the independent pilot investigation assessed a more aggressive infection, highlighted by the necrotic tissue present in the upper image. This was determined or classified as 100% infected. Subsequent to single treatment with Lunula, and following up with the toes 7 months later, the clinicians documented 100% nail clarity. Now, the nail on the left would indicate some degree of dystrophy, some degree of still hyperkeratosis. This will take some time to resolve, but it is important to understand or observe the change in appearance and color. The nail, no longer rife with mycotic discoloration, has shown dramatic improvement. But as the nail continues to grow in, it will take some time until the nail is beginning to grow more even and much smoother. Here we see the final image taken from the pilot investigation. This was a toe classified as being 100% infected, as evident by the yellow discoloration laced throughout the entire nail plate. Subsequent to single Lunula treatment, the independent clinics evaluated the toe and determined that it was 100% clear, free of disease. This is evident by the removal of the yellow discoloration. This again was absent or devoid of any adjunctive modalities, just a single treatment with the Lunula technology. The pilot investigation data provided enough evidence that warranted a FDA study evaluation under more rigorous and better defined protocol parameters. In the FDA study, 75 subjects were enrolled. This included 105 greater toes to be evaluated. Toes were divided into categories based on their initial percent nail clarity. So for instance, 37 toes had less than 25% of nail clarity. Another term, 75% of the nail was infected. 44 toes had nail clarity between 26 to 50%. And finally, 24 toes had nail clarity between 51 and 75%. Instead of a single treatment, 2 12-minute treatments separated by 1 week were applied. Again, no nail debridement was performed and no adjunctive therapies were used during this evaluation. Subjects were assessed at baseline and months 3 and 6. And at each evaluation point, the measurement in nail clarity was performed in millimeters, which was then used to calculate the overall percent nail clarity. And this was compared between baseline and months 3 and 6. When looking at the graph, we see not only change in millimeters, but as well as percent. At baseline, the average of percent or mean millimeter nail clarity was 4.6, which represented 43.4%. So what this means in this graph is that at baseline, the average nail clarity was 4.6 millimeters, or 43.4%. By 2 months, the nail clarity rose to 7.62 millimeters, or 62.9%. By 3 months, 9.59 millimeters was found for nails, and that's nail clarity, or 73.8% of the nail was now clear. By 6 months, 10.75 millimeters of clear nail was observed, or 79.8% of the nail was now clear. When we compare 6 months to baseline, we see that a total of 6.15 millimeters of clear, healthy nail has grown in. This is a 133% increase in nail clarity. This data from baseline to 6 months shows that Lunula, when delivered twice across all infected toes, successfully can induce nail clarity and produce a meaningful and substantial clearance in the nail. Across the entire study population, we can look at the total percent of subjects enrolled demonstrating some type of nail clarity. When we look at 76.2% of subjects, they observed or demonstrated a nail clarity greater than 25%. So that means 76.2% of subjects saw a mean change improvement in nail clarity of greater than 25%. 18.1% of subjects saw a nail clarity improvement between 10 and 25%. 2.7% of subjects saw nail clarity less than 10%, and 3% of test subjects saw no improvement. This demonstrates that out of the entire study population, 97% of subjects demonstrated new, clear, healthy nail growth, with 76.2% of subjects showing a percent change of over 25%. Here are some before and after images taken from the FDA study. Here we see a toe that was characterized as being infected of more than 75% of the nail plate. So this means that ultimately over 75% of the toenail is infected by the mycotic agent, as evident by the yellow discoloration. 6 months later, in the post-measurement, we see that the independent clinicians identified this nail now being 100% clear. This is identified through the removal of the yellow discoloration that was observed in the baseline or before image. This was again an outcome without any adverse event, and more importantly, no adjunctive components, showing the safety and overall clinical utility of the Lunula technology. In this image, the toe represents an assignment into the 26 to 50% category, with 26 to 50% of the nail being clear prior to treatment with Lunula. At 6 months, the nail was reported by 3 independent clinicians being 100% clear, without any adjunctive components or any adverse events. This shows a substantial clearance or improvement in overall nail quality in just 6 months' time following Lunula. Here we see the third and final image taken from the FDA clinical trial. This toe had an initial disease involvement or nail clarity between 51 and 75%, meaning 51 to 75% of the nail was clear prior to treatment with Lunula. After treatment with Lunula, the 2 independent treatments separated by 1 week. By 6 months, according to the 3 independent clinicians, 100% of the nail was now clear. This again, without any adverse event or any adjunctive components, demonstrates the clinical utility and safety of the Lunula device. The question, why Lunula, should have been answered through this extensive analysis of the clinical data that supports this multifaceted technology. There is a large and unfortunately growing population base throughout the world, with nearly 10% of the world's population suffering from this disorder. Lunula is the safe and proven solution to treat onychomycosis. We do not have the harmful anti-fungal medication side effects that ultimately limit or prohibit the use of these topical or oral solutions. We do not use the photothermal mechanism that other laser-based technologies have introduced into the market. Instead, we utilize the safe and subtle mechanism of photochemistry through the utilization of those 2 distinctive wavelengths, 405 and 635 nanometers. The Lunula device was designed and crafted to be a touchless, painless treatment, making no physical contact with the toe, requiring no nail debridement. This treatment and its delivery mechanism enables Lunula to treat the entire nail plate down to the nail bed, as well as the surrounding tissue, all the way down to the germinal matrix layer. This ensures for complete and total coverage of the mycotic nail. This treatment is also unmanned, allowing the technician to tend or address other issues within the clinic while the patient receives the treatment in the privacy and comfort of the treatment room. This technology is also designed to provide incremental independent revenue generation, high physician and technician and patient satisfaction, and provides a truly safe, no-risk technology. We would like to thank you for joining this Lunula scientific session. We hope that we were able to properly articulate the clinical utility of this multifaceted, innovative technology. If you feel that you still have questions remaining, please do not hesitate to contact your Prim Cogent representative. We would be more than happy to provide you with the necessary clinical literature and articles that demonstrate the utility of this technology. So with that being said, we would once again like to thank you for joining this scientific session, and we wish you the best.